For those of you that have been with me, one of the things I mentioned early on was that I'm not up here to hear myself talk. I actually don't even really care to hear myself talk. This is stuff I already know. It's important, though, that you know it, that you understand it, because I know what it did for me when I first got into the whole science thing 15, 20 years ago. I remember how emboldened I was. The more I learned about the science aspect, and how contrary it was to what I had picked up in college. Like everything you learn in college is Big Bang, it's millions of years, it's whatever, on and on and on. And you come out of college and I'm like, do we really need God? I mean, everything seems to work pretty well without him. I don't know why we need him. And I'll be honest, I was almost a bit timid to start looking into the evidence for a scientific evidence, if you will, for God. I'll be honest, if I'm being transparent, I was a little nervous about doing it because I was afraid that everything I had learned in college was absolutely correct, and if what I learned in college was absolutely correct, then the scientific evidence would not fall in favor of God. It would not fall in favor of the Bible. It would actually contradict it. And I was nervous about that. So even though I was a very lukewarm Christian at the time, I still identified as a Christian at the time, and thoughts started rolling through my head about, what do I tell my parents? How do I have a talk with them if it doesn't really turn out to be true? I didn't know exactly how I was going to approach that conversation. And as it would be, I didn't have to. The more I learned about the science side of things, the more I dove into the science side of things, the more I was inundated with the truth. And the truth always fell one direction. And it didn't fall towards naturalism. Naturalism being everything, the only thing that exists is the world around us and everything inside the universe. Naturalism is the same thing as materialism, which means only the material world exists. Nothing outside of the material world exists. So we have the natural, but there is no supernatural, which means no gods of any kind. But the more I looked into the science, the more I recognized that wasn't true. It screamed of a supernatural. It screamed of something more beyond us. And it kept pulling me in day after day. And I'm not kidding, every single day, every day for a few years, I read an article of some sort that was scientifically related, eventually turned into philosophically related, eventually turned into evidence for like the Bible and such, but something that would come under the umbrella of apologetics. Apologetics, it comes from the Greek word apologia, means to defend. And so Christian apologetics is to defend the Christian faith. And so the more I looked into it, the more it just kept coming out. It was as though you're only being told this much when you're in high school and junior high and especially college. They give you this much information. And they shield you from all the other stuff that's out there. And as I recognized that, I became a skeptic, which is funny because atheists always refer to themselves as the skeptics. The truth is, I find myself to be way more skeptical than they are. I'm very skeptical now. I'm like, is that really true? I don't know if that's true. I think I'm going to look that up myself. And it's what's led me down this path of truth and to the place that I am right now. So over the last 5 weeks, we had last week off, but with the last 5 weeks, we've discussed a lot of things. I'm going to throw some stuff out there. If you're new tonight, you will maybe not know the answers to these. If you are not new, you should know the answers to these. Otherwise, I have been wasting my time on you. Stop wasting my time on you. In the first week, we talked about 2 different kinds of science. Can you guys remember the 2 different kinds of science we talked about? You can, go ahead. Can you remember? Can anybody remember we talked about 2 different kinds of science? Historical and empirical science, right. So creationists and, let's say theists and non-theists will mostly agree on which kind of science? Empirical science, right? Because empirical science is 3 things. What are those 3 things? observable. Testable, observable, repeatable. Now, if you're new tonight, or that if you hadn't heard that before, please remember that, because I'm gonna bring up empirical science tonight. I'm gonna talk empirical science. When I say empirical science, it means science that is testable, observable, and repeatable. Now, when it comes to those kinds of things, we as creationists and those that are not creationists, we really shouldn't have much of a problem. We might dicker a bit on how to interpret that stuff, but really there's not a big basis of issue there. Where our bigger issue comes in is with the historical science. And that stuff is not observable. It happened millions of years ago. Well, no one was alive millions of years ago to observe it, so it's not observable type of a thing. How do you test for millions of years? Someone would have to be around for millions of years to test for millions of years, so it's not testable. So there you can see the issues that come into empirical science and historical science. Where we will have an issue with others will be in the historical side of the science. What we'll talk about tonight will be both empirical and historical, so when I bring those up, just please keep in your mind what those are and the differences between the two. That'll make a difference. We also talked about DNA. DNA has a language. That language is made up of 4 letters. What are they? A, C, T, and G. That's what you should have said. Those 4 letters combine with one another. What do they combine? A combines to T. C combines to G. Very good. Okay, excellent. And then we learned about a process within the DNA, and it was called DNA error correction, right? DNA error correction. And that was the genome recognizes what is and isn't an error, and it knows how to fix those errors, right? Now, we thought about it, and we came to a conclusion that the only thing in the world around us that knows A, what an error is or isn't, and B, how to fix that error is what? Intelligence, intellect. That's absolutely right, right? It always comes back to an intelligence of some sort. So we've done a nice little recap. I could go into more, but I wanted to just refresh your brains, okay? These last few weeks haven't been just about presenting this stuff to you. It's about you guys retaining it and then knowing it, and maybe being able, even if you can't like talk in depth about it, that's okay. If you can just drop a nugget every now and then to people. You know, well, someone might say something to you, well, DNA shows common ancestry. It proves that we evolved from lower primates. And a response might be, well, what are your thoughts on DNA error correction then? How does that fit into the evolutionary world? And that's a good question to ask. It puts the ball back in their court, but at least it gives you a ballpark of something to talk about with them, and maybe it puts a stone in their shoe, and maybe they go home and look that up, and maybe they go, I'd never heard that before. That's really interesting. And that's what we do as apologists. Our job is not to convert somebody on the spot, turn them into a Christian, right? Yeah, our job is just to witness. Our job is to give them the gospel message, period, end of story. You cannot turn them. That's the Holy Spirit's job. And I love that. Why? Because it takes all the pressure off me. All I have to do is tell the people the gospel message, and then let the Holy Spirit do his job. And I can go back to doing mine. So, with that, let's get into tonight's discussion, dinosaurs. So dinosaurs are something that has been brought up in the past. I get this quite a bit. Kids obviously are into the dinosaurs. They like to learn more about it. But there are adults too that I get tons of questions from. And those questions are usually around, well, like how do we reconcile dinosaurs with what we read about in the Bible? Can we? Is it possible? Do we need to? If we can't, do we even need to? Is it necessary to show that the Bible is true and that God exists? Well, we're going to touch on a few things tonight, and what I will do is present the evidence to you and allow you to kind of come to your own conclusions on it. But this will be stuff that is out there in the literature. Nobody ever talks about it. I shouldn't say nobody ever talks about it. There are tons of people that talk about it, but if you don't follow it on a regular basis, you're not in that loop. You're probably not talking about it with them. So I'll bring this to you tonight, and you guys can do with it what you will. But let's approach these dinosaurs. So dinosaurs, according to science, dinosaurs died out 65 million years ago. Period. End of story. For them, and what I mean by them is academia, professors, PhDs, especially those that are non-religious, but even religious PhDs, but non-religious PhDs, they will all contest dinosaurs died out 65 million years ago, period. Giant asteroids hit the Earth, there was a bombardment, it kicked a bunch of dust up into the air, the dust covered the light from the sun couldn't get through, hence the plants and such couldn't grow. When the plants couldn't grow, then the larger dinosaurs that needed the plants died, then the larger dinosaurs that needed the plant eaters, they died as well. There you go, the end of the dinosaurs. So in a very quick synopsis, you also need to understand the direction that we're coming at this from, and that is dinosaurs died out 65 million years ago. That is just well understood in the scientific community, and it's an absolute no-brainer to them. They are literally dogmatic in their stance on this particular point. There are many other things that are like, well, you know, we don't really know about this, we don't know about that. But when it comes to dinosaurs, they are very dogmatic. They died out 65 million years ago. So keep that in mind as we push forward. Empirical science, historical science, and dinosaurs died out 65 million years ago. Now, we'll have to do a little bit of science prior to getting into the deeper stuff, but if you don't understand the science I'm about to present, it's going to lose you for the rest of the talk. So let's just talk very briefly about a few things. Number one, my clicker isn't working. Tony, where are you?
Is it turned on in your hand?
It is turned on, yep. Are you plugged in?
Yes, I plugged in.
Unplug it and plug it back in again for me and see if maybe it makes a connection there.
All right.
I think it's, ooh, that's a good sound. That's the sound I want to hear.
Is that a good sound?
That is a good sound. But it's still, it's a terrible sound. That's a terrible sound. This thing shows it might be connected. There we go.
Oh no, that was me.
Did you do that?
I did that. I'm sorry.
Okay, so don't touch anything. Let me see if I can move it. Oh, hey, there we go. Hey. Okay. All right, we're in.
All right.
Okay, human collagen. This will come into play tonight. Collagen is the most abundant protein in the human body. It's regarded as a substance that holds the whole body together. It can be found in bones, muscles, skin, organs, hair, tendons. Ladies, you ever bought some sort of collagen product of some sort? Most of the women will probably have heard of that. If you've never purchased one, you've at least heard of that. So collagen is the most abundant protein in the human body. And it gives skin its strength and elasticity. It's also responsible for the replacement of dead skin cells, yada, yada, yada. Just keep in mind that we're working with collagen in the human body, so human collagen. The next thing you need to understand is that we also have soft tissue in our bodies. Soft tissue is basically anything that's not a bone. A bone is hard, right? So everything that's not hard, like your bones, is considered soft tissue. So that includes your tendons, ligaments, skin, back muscles, nerves, blood vessels, just anything you can squeeze on your body is considered soft tissue. Now, here's the interesting thing about soft tissue and collagen, and that is when an organism dies, there is nothing left to sustain the soft tissue, so it slowly starts to decay. This obviously is common sense. We see it around us all the time. You go to one of the lakes and the dead fish, might be a dead fish sitting in front of you, right? And it's already starting to deteriorate. We're slowly seeing the effects of the decay on the soft tissue. We're seeing the effects of decay on the collagen. That's a real life example of that. A dead bird that the neighbor's cat may have killed. Same thing, right? Flies are all over it and whatnot. What you're seeing is the decay of soft tissue. Now, what's interesting about all of this is that these rates can actually be measured. We can take this soft tissue, we can then put it in a laboratory, and we can measure the decay rate at which it dissolves. We can expose it to water. Does it dissolve faster in water? We can maybe put it in some sort of formaldehyde, which may may hold up the collagen and the soft tissue a little better than the water will. Do you understand that kind of premise behind it? So this is very important because this is empirical science. Testable, observable, and repeatable. We can take any dead organism and we can test its decay rates, and we can do it all over again with different organisms, frogs and whatever else. So these rates can be measured. So once again, the best part is that they can be observed. This is stuff that we can actually see in a lab. And we can then track these rates so we know how quickly or how slowly these organisms eventually decay away. So we have organisms that die in today's world. We watch and measure these rates. And this cannot be done with other types of organisms. So something that can be observed, like this, decay rates of collagen and soft tissue, cannot be done with evolution. Evolution in terms of, let's say, a single cell into a human, right? A lower primate into a human. That's never been observed. We've never observed any of that happening. So what's nice about this particular process is that it can be observed. Put it in a lab, observe the decay rates, track it, test it, do it all over again. So here is an article that was printed not too long, well, it was maybe 20 years ago or so. But this particular article went out and said, well, let's do that. Let's just measure the decay rates of these different substances and let's track it. Let's just see if we can get at least a baseline for how fast or slow things decay. So they went out and they did this paper, and in this paper you can see that they came up with a chart. So up here we have DNA, collagen, right? We just talked about collagen. And osteocalcin, we will not talk about that. It's of no importance tonight. Just look at the DNA and the collagen. Then over here, if you track it, this will give you decay rates at different temperatures. 0 degrees, 10 degrees, 20 degrees. And then one more note is up here, your detection limit is in years times 10 to the third, 10 cubed. For you math geniuses out there, 10 to the third is how much? It's what? 1,000. Who said 1,000? Nice, smart, nice. Okay, so 10 to the third is 1,000. All right? For those of you that got it wrong, don't tell your neighbor. Just pretend like you got it right. Give yourself a pat on the head. So these rates are times 1,000. So what you're going to do is any number up there, just put 3 zeros behind it, which will make it super easy. And that'll give you the actual number. So if we look at DNA here, and we go over at 0 degrees, DNA should last about 125,000 years, right? And then what happens is it decays to the point where you can't detect it anymore, okay? So at 10 degrees Celsius, it's 17.5. You can't see that very well, but it's 17.5, which would be how many? Okay? And then 2.5 gives us 25,000. Okay, great. So this kind of gives you a ballpark of what we're talking about. And obviously, you can see by the chart that the warmer it gets, the quicker it decays. Duh. I mean, right? Go to the store and buy some beef. Buy 2 steaks. Take them home, throw one on the counter, and just leave it there. Throw the other one in the deep freeze. Which one's going to last longer? The one in the deep freeze, of course, right? Because it's cold and it holds all of the collagen and soft tissue intact. It preserves it, correct? But when it gets hot, it breaks it down. And it breaks it down so much quicker than it does in a cold environment. Now let's look at collagen. So when we look at collagen here, estimated rates, you have 2,700. So at 0 degrees, take this number times 1,000, and that gives you 2.7 million years, right? That's what you should get. So 2.7 million years. Look how that changes as we get to the warmer. At 20 degrees Celsius, it's only 15,000. How cool is that? Like, that's a huge difference, right? And that's in the change of temperature down here. So at 0 degrees Celsius, these are all in Celsius. 0 degrees, we're in America, so let's do it the right way. Let's make it Fahrenheit. 0 degrees Celsius is 32. 10 is going to be about 50 degrees Fahrenheit. And then 20 is about 68 degrees Fahrenheit. Okay? So quite a difference from holding something at 32 degrees to bringing something up to 68 degrees. Where are you going, Tony? What are you doing? Hey, crazy boy. I'm about to switch slides, dude. It's not going to be of any consequence here in a second.
Have your way.
So, what we've seen here is an article on the decay rates of DNA and collagen, right? Now, in science, this is how it works. If new evidence comes out, then you have to work off of the newer evidence. And there was new evidence that came out on DNA. Now, I want to make something clear. Keep in mind that DNA and collagen, which is soft tissue, right? Well, collagen's in bone too, but when we're talking soft tissue and we're talking DNA, they are two separate things. So DNA and soft tissue are not the same things. They are two separate things. And they are affected differently by the environment. So keep that in mind pushing forward. We have the collagen rates. I want you to pay special close attention. If you're taking notes down here in front, write down the collagen stuff. Okay? Now, I want you to also keep track of the DNA. This was a newer paper. This was a newer paper that came out. And it's on the DNA decay rates. This was the most extensive study ever done on DNA decay rates when it came out. And they looked at 158 moas. Anybody know what a moa is? It's like an ostrich. It's an extinct bird, but it was big like an ostrich. So they found a graveyard of like 158 of these dead birds, right? And they were able to reconstruct the genomes, and they looked at the birds themselves and were able to measure the decay rates in these birds. And they did it on a half-life. Raise your hand if you understand what half-life is. Okay, not all of you understand half-life. This is how half-life works. Let's say we've got 100. Now the half-life of DNA measured out to 521 years. That's the half-life for DNA. What does that mean? That means that if you start with 100, after 521 years, you will have half of that, which is what? And in another 521 years, you will have half of that, which is what? And then 521 years, you have half of that, which is 12.5, so on and so forth, right? So just like, let's pretend it's dollars. So 100 pennies to 50 pennies to 25 pennies to 12.5 pennies to 6.25 pennies to 3.125. Okay, you get it, right? We eventually get to a point where you can't even measure the penny. Like it's less than a penny. It's of no value. This is how it works in DNA. We get to a point in DNA where the half-life reaches a point where we can no longer find the DNA. We can't measure it anymore. It's just decayed to the point where we just can no longer find it. Okay? So they measured all of these. They figured out that the half-life of DNA is 521 years. And here's the chart on DNA. So don't have to take these by 1,000, yay, right? This is nice. So here you have different temperatures, 25, 15, 5, and -5. And then over here, you have your time until you reach one base pair is what that means. It's basically, you reach a point where you just can't really detect it anymore. It's so gone that you can't detect it. So at 25 degrees, you'll see that DNA will only last about 22,000 years before we cannot detect it anymore at all, period. And the colder you get, the longer that gets. At -5 degrees, though, you can get DNA to last an estimated 6.8 million years. And so what they do is they measure the rate, and then they just extrapolate that backwards in a process. They call it uniformitarianism. But it's a process where they figure out what the rate of decay is, and then they can extrapolate that out in the past to get an idea. Obviously, no one's been around for 6.8 million years to figure out if DNA at -5 degrees will be persistent or not. But this is what they do. They can extrapolate that out. Keep in mind that the science is still built on empirical science. They are testing, observing, and doing repeatable type science on actual DNA. So this was the most extensive study ever done on DNA decay rates. Okay, phew. All right, so are you still with me? Soft tissue and DNA, not the same things. Soft tissue decays at one rate, DNA decays at another. Are we all on the same page so far? Okay, very good. Okay, so just a little bit, just a tiny bit more, and then we'll keep pushing forward. So there's a place in Montana called the Hells Creek Formation. You can see where it's located here. Right here in the Montana area. And this is the Hells Creek Formation. So it's got all kinds of fossils, and you can take tours up there. And they'll take you out on a fossil hunt, and you can look for fossils and hunt for fossils. It's actually it's a hoot, right? I would love to go on something like this. I've been told by many people that have that it's an absolute blast. So if you ever get a chance, want to do something fun, go out and hang out in the hot sun, digging through dirt, and maybe you find something, maybe you don't, but hey, what's a week, right? No, that's not my idea of a week of fun, but hey, you never know. Never know what you might find. So, in the Hells Creek, there's a bunch of fossils. And this is established science. I want you guys to understand that what I showed you here is all from peer-reviewed secular research. So these guys that did these papers, this is from the Royal Society, which is one of the most prestigious journals in the world. Why is that a big deal? If you're a scientist, the biggest thing you can get is published. Once you do a master's or a PhD and you write a paper, it goes through a peer-reviewed process, and your peers review it, and they're just diligent, and they're really heavy-handed on what they hit you on, and you redo it, and you redo it, and you redo it, and if you make the cut, eventually your paper will be published in one of these major journals, and that is a big deal, right? That's like a triple gold star in class. That's huge. People are excited about this. So this was put into one of the most prestigious journals in the world, peer-reviewed. Same thing with the last one. That was actually just an article. Articles aren't as diligent in terms of what they have in them, but this is. So keep in mind that this information all came from peer-reviewed journals. So in the Hells Creek Formation, they have a bunch of fossils, and this was established science. We know the decay rates of DNA. We know the decay rates of soft tissue. It's not a big deal. Nobody cared until they found soft tissue in a T-Rex bone.
Oh.
And that was a game changer. So a lady by the name of Mary Schweitzer was out on a dig in Montana at the Hells Creek Formation. And she came across the bone, actually. You're going to see a video on it. There was a gentleman who stopped to take a lunch break. And he saw up on this huge mountainside. So imagine you're standing there and there's just this huge cliff of dirt and rock and such, right? And he notices up here, there's like a bone thing protruding. Something's there. Like, he knows it's something. So he gets up closer, and the more he starts looking at it, the more he recognizes it's a bone. It's an actual bone of some sort. So they start digging away at it, and digging away at it, and digging away at it. And what they find out is that it's a T-Rex thigh bone. Well, they have an issue. This T-Rex thigh bone is 50 feet down from the top of this rock wall. 50 feet down. And buried right in the middle of this sediment here. Right? Just in the middle of this rock wall. So they've got people rappelling down and jackhammering. Think of Mount Rushmore, right? They've got people rappelling down this rock wall trying to dig out this dinosaur bone. And when they dig it out, they of course don't dig around the bone itself. They dig a huge swath around it, right? You got to kind of dig a big spot around it. And so they dig this thing out. They're in the middle of Montana. And now they're like, okay, now what? And they don't know what to do with it. Because they couldn't lift it out. It was too heavy to pick up and put on a truck. So they were going to bring a helicopter in, but it was too heavy for the helicopter to take out. It was huge. And it's in this big stone, you know, it's in this big rock matrix. So what do? Mary Schweitzer calls up the other guy on the dig. Jack Horner is his name. Jack Horner is the guy that they talked to for all of the Jurassic Park and Jurassic World movies. So when they needed experience on what about this dinosaur? What about the behaviors of that dinosaur? The producers of the movies called Jack Horner. He was the man. So Mary Schweitzer calls up Jack Horner and says, what do I do? And he's like, well, cut it in half. She's like, what? Wait a minute, what? Because when you find an intact thigh bone of a T-Rex, you don't cut it in half, right? It's intact. And he's like, I don't care. We got to get it back to the lab. Just cut it in half. All right. So they cut the thing in half. The helicopter comes in. It pulls out both pieces. They get it back to the lab. So now what do we have? We have a thigh bone that should have never been touched, but now it's been cut in half. And so Mary Schweitzer takes bits and pieces of the bone fragments, because when you cut it in half, it's not perfect, and you get little bone fragments that kind of chip off. If anybody's ever, used a chainsaw, you know how that works. You're cutting through the wood, and sometimes you get little pieces that fly off or whatever. Same thing with the bone. And she puts it in this really, in what's acid. But when we think of acid, don't think of the stuff that you see in the Batman movies where you burn down to your bones or anything. It's a really, really, really weak acid, and all it's meant to do is just remove kind of some of the rock and stuff around the actual bone itself. Well, somehow Mary got the concentration a little bit too strong, and it actually ate away not only the rock, but it ate around all of the bone material as well. Now in fossilization, what happens is, the process is, minerals replace the bone. So when you find a fossilized bone, what you have is basically a rock. It's just been replaced by minerals. You guys are all nodding, right? You're with me? Yeah, that's what happens in fossilization. So an animal dies, it's quickly buried, it has to be water, it has to be sediments like a sandstone or a limestone. The minerals get into the bones, they replace the bone material with the sandstone and the minerals, and then it hardens, and so you have something that looks like the bone, but it's actually just, it's minerals and stuff, right? Well, when she put this in acid, it ate away the rock, and it ate away the minerals that were there. Well, when you do that, you have nothing left. I mean, in a really old fossil, you have nothing, because it was all minerals, right? Well, she noticed a problem. That was left behind. This is T-Rex soft tissue. So what started out as a completely arbitrary find in the side of a rock wall, they couldn't move the bone, so they had to cut it in half, which is unheard of, but they had to do it. And only because they cut it in half did they get some bone fragments that she was able to put in a little petri dish and dissolve everything away from it, expecting that, oops, I got the concentration too high, nothing's going to be left. But what's that? Puts it under a microscope, this is what she sees. This is T-Rex meat. So this is actual soft tissue from a T-Rex. Well, she tells the person helping her, she's like, no, impossible. Why did she think it was impossible? Because dinosaurs died out 65 million years ago, period. And empirical science, not historical science, empirical science has taught us that DNA and soft tissue does not last 65 million years. Even under the best conditions, if we go back to that original article, you'll find that soft tissue only lasts maybe 1 million years. It was well understood in the scientific world. She recognized it immediately. She was looking at blood vessels, and she knew they were blood vessels, but her brain wouldn't allow her to believe they were actually blood vessels because she's been trained that dinosaurs died out 68 or 65 million years ago, period, done. However, she has this in front of her. So she tells her assistant, I don't believe it, do it again. So he did it again. What did they find? Same thing. I don't believe it, do it again. What did they find? I still don't believe it, do it again. They kept pulling fragments from this T-Rex bone. 17 times she ran that experiment. Testable, observable, repeatable, empirical science. This is how it works. 17 times she ran that experiment. Every single time she got soft tissue out of every single fragment she looked at. So she said, well, this is going to cause a storm. But she published her results anyway. This is the paper. She was lit up by the scientific community. They were like, absolutely not. What you found was contamination. It was contaminated by something else. Maybe skin cells from your fingers because you touched the bone or something, right? No way. You did not see what you think you saw. Okay? These are not the droids you're looking for. So, a lot of people thought they were biofilms, like those blood vessels and such. They thought they were called biofilms, which is like a bacteria, right? And she was absolutely lambasted in the scientific world for printing this stuff. Because everybody knew, not just her, everybody knew soft tissue does not last that long. Period. End of story. And so, of course, she had to be wrong. Without doubt, she had to be wrong. Well, I don't know this for a fact, but I kind of think this is true. I think Mary said, all right, fine. Don't believe me? That's fine. I'll go find another one. So she went back to Hell's Creek. She found a hadrosaur dinosaur, which is a duck-billed dinosaur like you see on Dinosaur Park. She found a duck-billed dinosaur. She cut it open. She took every single precaution so that they couldn't claim contamination. When she found the bone, took it back to the lab, cut it open, guess what she found? Soft tissue. Ta-da. Yeah.
No one ever done this experiment with a fossil before?
So, I'm going to touch on that in a second. So let me tell you this. Let me tell you this. No one at this point had ever popularized this idea before. Okay? So of course, this blows up. Mary Schweitzer's onto something here. She found a T-Rex. The T-Rex had soft tissue in it. She goes out and she finds another one, a hadrosaur, opens it up, soft tissue. She's like, ha! Right? And of course, now the scientific community has nothing to claim. They can only say, yeah, T-Rex was soft tissue. They didn't want to. Why did they not want to admit that there was soft tissue in these particular bones? It changes everything. It is a game changer if they admit this stuff. This one was 90, I think the first one was 97. This one was 99, I do believe. This shows me May of 2009. How about that? And I think the first one was 97. It tells me at the bottom, but because I am old and blind, received on 2008, published in March of 2009. This one here, I'm missing it. It's in here somewhere. Oh, March of 2005. So the T-Rex was found in 2005. The Hadrosaur was found in 2009. They both came back with soft tissue in them. So now they're stuck with a conundrum, right? But I'm going to let you hear about this from Mary herself. They did this on 60 Minutes. I'm going to show you about a 2-3 minute little blurb here. If you want to look it up, the whole video is about 14 minutes. And you can watch it for yourself. I'm tuned to this, so I hear certain things when I listen to scientists talk. So I'm tuned to certain things. I want to see if you guys pick it up too. But I want you to listen to this. Hopefully the audio will come through, and Tony's going to make that happen because he's a miracle man. So let's see if we can get this going.
What happened next happened by mistake. Mary put some fragments of the bone in acid to dissolve away the outermost layer of mineral. But the acid worked too fast, and all the mineral dissolved away. Being a fossil, there should have been nothing left. But there was, and it was elastic, like living tissue.
This is the piece.
No. She showed us video she took under the microscope.
That's really what happened?
Yes.
That's the dinosaur
Yeah,
bone?
without mineral now. That's what was left.
It looked like the soft tissue she would have expected to find if it had been modern bone. This was impossible. This bone was 68 million years old.
So you see this, and you think, what? You
I
see,
didn't want to tell anybody.
that you'd be ridiculed,
Yes.
right?
And so I said to my technician, okay, do it again. I don't believe it.
And yet, in sample after sample, they were there. Things that looked suspiciously like flexible, transparent blood vessels. She finally mustered the courage to tell Jack.
She said you dissolved the bone away and there were blood vessels. And, you know, I was like shocked. I mean,
How could that be?
How could that be? That's right.
The things Mary was finding inside dinosaur bones,
Look at that.
blood vessels, and even what seemed to be intact cells, pose a radical challenge to the existing rules of science, that organic material can't possibly survive even 1 million years, let alone 68 million. Mary, Jack, and their team published their B-Rex findings in a series of papers in the journal Science and were promptly attacked. Critics said their samples might have been contaminated, or that the supposed blood vessels were actually something called biofilm, a type of slime. But as Mary showed us, she's been able to replicate her findings. These are pieces of an even older dinosaur, a well-preserved 80 million year old duckbill. When she dissolved it away in acid,
Let's put this under the scope here.
well, look, is that a blood
This
vessel?
is a blood vessel. You see the branches right there? And look at all of them. And it's so consistent over and over and over again. You do this bone and it comes out and I get excited every time. I can't help it. I mean, 8 million years old.
Mary published her new results last year, and while some of her critics have been swayed, the controversy still rages, and the stakes are high. If blood vessels can survive 80 million years, what about DNA?
So, Mary says, I had him do it again. Because there's no way this should be possible. I'm putting in subtitles, okay? I told my assistant, do it again. Why, Mary? Because it's impossible. We know, based on empirical science, that soft tissue does not last that long. So I told him, do it again. She did it 17 times, kept getting it over and over and over again. Then she found the hadrosaur, did it over again. Did you guys pick up on a few little things in there? Did you notice what they kept saying? This bone is 68 million years old. Well, this bone's 80 million years old. So here's the situation. I'm all about trying to figure out how soft tissue could last 80 million years, 68 million years, and they've already put out ideas on how they think that could have happened. Mary Schweitzer herself has written a paper about how she thinks maybe it might have been able to last that long based on iron in the blood, so on and so forth. However, and that's totally scientific. You guys need to understand that a scientific question is to ask, how can soft tissue last for 68 million years or 80 million years, or they found like 110 million year old stuff. I'm reading papers now where you guys will appreciate this from last year, or from last week, 2 weeks ago, sorry. They're finding stuff in Cambrian and Precambrian stuff that still has soft tissue in it. Precambrian stuff goes back 550 million years. And they're finding soft tissue in it. So, back to the dinosaurs. A scientific question is absolutely to ask, how can soft tissue, or can soft tissue last 68 million, 80 million years? What question are they not asking? The question that is not being asked, and it's every bit as scientific, is maybe these bones aren't as old as we think they are. That question is every bit as valid as the other question, and it's actually the bigger elephant in the room because we have actual empirical studies on decay rates of soft tissue. We know soft tissue doesn't last for millions of years. We are in a quandary. Either the empirical evidence is wrong, or the scientist's idea of what happened 65 million years ago could be wrong. We're at an impasse. So this is what we see in science. They know this stuff. What I'm giving you here is peer-reviewed science, right? Mary Schweitzer herself is telling you, yeah, it's soft tissue. Look at it. I mean, it's squishy. It's like springing back, right? How crazy is that? It's literally meat from a dinosaur. Do you guys know what this is? It's a big fish. Have you guys seen Jurassic World? Have you seen the part where he comes out and back in the thing? This is called a mosasaur. So, guess what? This is what happens when scientists get dogmatic. When scientists decide things died out 65 million years ago, period, we don't even need to discuss this anymore because there's no reason to discuss it anymore. Not only is that a science stopper, but it doesn't help us push forward in the world of science. Science is all about trying to falsify everything that's ever been found. That's what science is all about. It's about pushing forward. It's about following the truth no matter where it leads. That's what real science is all about. When scientists get dogmatic about how they think, they are now no longer true scientists. They're closed-minded. They're not objective anymore. That's not what science should be about. And it's a science stopper. Now that they found soft tissue in a T-Rex and a hadrosaur, guess what everybody's looking for when they dig up bones? Soft tissue! Now everybody's looking for it. We could have been doing this for decades. And nobody came forward and said, you know what? What if we're wrong about the 65 million year old thing? Nobody's asked that question. Nobody's challenged it. Which is how science works. It's all about challenging the guy next to you. And then redoing the experiments to see if it actually comes back as valid. So this mosasaur was found. And on this mosasaur, they found the remains of the animal's last meal. This particular specimen was a fish-eating mosasaur. It also comes from soft tissue still preserved, which does not happen very often, Martin said. Well, duh. Unless you're looking for it, then it happens all the time, because now we're finding it all the time. There is cartilage still on the shoulder blade and on a bone called the coracoid. So, and actually, this is kind of cool. I like this example of the mosasaur. Why? Because if you guys don't mind driving for a little while, you can actually see this specimen. Do you know where it's at? School of Mines. I had the lady at the School of Mines give me and some students a tour, and she took us in the back room and she showed us how they extract the bones from the rock and the matrices and the casts that they make and blah blah blah. And at the end of the tour, she showed us this because I asked her. I said that we want to see that, so she kind of saved it for the end. Well, this is in the back room. They may have pulled it out now. It's possible that it's on display down there, but this is down at the School of Mines and Technology. Now, if you're in the building where the museum is, there's another building back behind it. I think it's the paleontology building back here. and it's named for this guy, and I think his name is Jim Martin. So it's like James Martin building or something. You'll see it back behind the museum part. It's in the back room of that building back there. And I've got pictures of myself with it because I wanted to prove to people I really did see that before they like destroy it or send it off to someplace else. But he claims he found soft tissue on this thing, and that was right here in South Dakota. He found that specimen in the Chamberlain area. Yeah. So we have an example right here at home of a mosasaur that's supposedly like, I think he said, 80 million years? So once again, 80 million years is what they're pushing, but they found soft tissue on this thing. Now, the next step was then, they started doing deeper looks into this stuff. Do you remember the pictures that we saw in the video? And she was like, look at the blood vessels, and they're stretchy and squishy. Well, now they started diving deeper into it. And this was a paper that was released in 2012. So we have 2005, 2009, 2012, right? Molecular analysis of dinosaur osteocytes support the presence of endogenous molecules. Man, I sound so smart. Okay, listen, scientists like to impress each other with these really, really big words, but let me break this down for you. So they did an analysis under a microscope of dinosaur osteocytes. Osteocytes are bone cells. So osteocytes, they're just bone cells. And it supported the presence of endogenous molecules. Endogenous means it belongs to the specimen you're looking at. It didn't belong to contamination. So if you have a fossil, if you have something here, and let's say like a bird dies above it, sometimes the bird decay can leak down into the thing below it, and you get contamination. When you study this thing, you're getting all kinds of results, but what you're really getting is the decay of a bird that was dead above it. Does that make sense? Okay, so what this is telling you, though, is that these osteocytes, these bone cells, are endogenous to the actual specimen, which means they don't belong to anything, or they belong to the actual specimen itself. So these bone cells are actually the bone cells from the dinosaur itself. We've taken the next step. So now, not just finding soft tissue, now we're finding actual cells that we can pull from the dinosaur itself. Immunological and mass spectrometry evidence for preservation of proteins comprising extant osteocytes, yada, yada, yada. These data are the first to support preservation of multiple proteins and to present multiple lines of evidence for material consistent with DNA in dinosaurs, supporting the hypothesis that these structures were part of the once living animals. So, what they're talking about here, do I have the picture? Yes, I do. This is from the 60 Minutes. Do you remember these things here? These are osteocytes. These are bone cells. They're branching off. They have all the little branches. Here's what I want you to pay really close attention to, because this is kind of super cool. These things right here, do you see these really big dark spots? Do you see those? Does anybody know what those would be inside a cell? That is a cell nucleus. And what do we typically find inside of a cell nucleus? DNA, that is correct. And the way they tested that was they put in these antibodies. And these antibodies only attach to certain things. Then they look at it and the color of it, and they color these antibodies in green or purple or red or something that stands out, right? And they shoot it into the cell, and then they look at it again, and if it attaches to anything, it lights up. Well, it did exactly that. They shot these antibodies into the nucleus of this thing, and it attached to what they can only guess is DNA, because it's the only thing these antibodies attach to, period. And they found that inside these nuclei, not only were they finding cells, now they're finding nucleuses, now they're finding DNA. Now if we come full circle, do you remember what I told you about the decay rates of soft tissue compared to the decay rates of DNA? How did DNA do against soft tissue? Not as good, right? Not as good. So now we've taken the next step. Now not only are we finding soft tissue, but we can close the gap. So we could say that 65 million years Based on the evidence, I don't know. So let's move it down to where we have for a number to work with. And now we can move that even closer, right? Because now we can find, now we're finding DNA. So this is going to take us back to your question earlier, right? So it wasn't that they didn't publish it, it's that they didn't popularize it. However, here is an actual peer-reviewed paper that was done back in 1994. And the paper is a DNA sequence from Cretaceous period bone fragments. What does that mean? It means he found a dinosaur, he found stuff in the dinosaur bones, and he went as far as to sequence it. What does that mean? What does it mean to sequence stuff? You guys ever seen that 23andMe commercials and those genealogy commercials? You send in your blood and they look at it. They sequence your genome, which is a bit scary to me at this point. Big brother type stuff, but hey, not here for that. But they sequence your genome, and what they can do with that then is try to tell you, well, it looks like you came from the southern regions of Italy or whatever. So you're part this, you're part this, you're part this. I did that, and it came back that I was mostly shepherd and part Siberian. I don't know what that means. Evolution took a strange turn with me. Anyway, this guy did it. So this guy did it. This is back in '94. And this is what he came up with. ACs, T's, and G's. He actually pulled nucleotides from the actual sequencing. The sequencing came back and gave him actual letters from the dinosaur. So this was already out there, right? So this is very curious because this was already in the literature. It's just nobody was talking about it. Mary Schweitzer was the one that popularized it when she found the soft tissue in the T-Rex. The truth is, she didn't find it. It was already found. This guy had already done it. It had been in the peer-reviewed literature for quite a while. And this is the paper.
They didn't link their paper.
Yeah, well, they didn't talk about him. You won't find that they ever talk about this guy. They only talk about Mary Schweitzer and now what they've done is they fully accept that it is, period, end of story. It is soft tissue. It is from the dinosaur itself. What they will not accept is that it's anything less than 65, 68, 80, 110 million years old, period. They will not ask the other question. The bigger elephant in the room, none of them are asking this question. Not in public. They might be doing it behind closed doors, but they ain't doing it in public. Yeah, they found this guy wearing cement shoes in the bottom of the Hudson. He was scheduled to speak the next day, so science got rid of him. I don't know. I think he may still be around. Yeah, this Scott Woodward is not ever spoken of, but this paper is still out there. You can look it up. Oh, hey, as a matter of fact, since we have a little time and you guys are you know how you usually red herring and everything? That means just kind of drag me off in different directions. I get to drag you off in something. This is funny. You guys will find this interesting. So this guy supposedly found DNA and he sequenced it in these dinosaur bones. Here's something that you guys will get a kick out of. If I can get back to it. There was the paper way back when. I want to go back to it anyway. Tony, you up there?
Yes, I'm
Can you
still
hit the
here.
up arrow for me?
Hit the back arrow?
Yeah, the up arrow or whatever. I want to go up some slides. I want you to take me back to the paper on the half-life of DNA at 521 years. Yes, that one.
Okay.
Yeah, that one. No, go down one, right here. Okay. This is really funny. So when I was reading this paper, this stood out to me, and I'll make it stand out to you because it's kind of funny. So in this paper, right here, starting here, however, even under the best preservation conditions at -5 degrees, our model predicts that no intact bonds at one base pair will remain in the DNA strand after 6.8 million years. This displays the extreme improbability of being able to amplify a 174 base pair DNA fragment from an 80 to 85 million year old Cretaceous bone. Notice how it's 1? Notice how the footnote is 1? If you go down and look at what footnote 1 is, it's the paper I just showed you where he sequenced actual thing. So this guy knew that the Scott Woodward paper was actually out there. And he said, after our studies, even at -5 degrees, it will only last 6.8 million years, which means this, basically what he's saying is, this Scott Woodward guy is wrong. He totally didn't sequence that. It must have been contamination. It had to be something else. There's no way you could pull any nucleotides from something that old. So he tried to kind of quietly discredit his paper, right? And if you're in the thing, like when I read that, I'm like, ah, that's funny. Most people will never ever get that, but that's what he's doing. He's trying to discredit that paper from '94 where the guy actually sequenced the dinosaur stuff prior to all the big stuff coming out, right? So this was kind of a jab at him. He didn't need to put that in. There was no reason for him to put that in. He stuck that in on purpose as a purposeful jab towards Scott Woodward saying, listen, you did not find DNA sequences.
How do they get past the just common knowledge for today that if you don't have cold, you can't even begin to approach saying that this would last all those years, especially in simply a rock matrix out in the Badlands, basically, where it doesn't stay cold for a long time and it doesn't even approach that kind of cold. So where do you get tissue preservation, or for that matter, DNA preservation, without the lack of cold constant?
Well, let me ask you this. Remember when we talked about DNA error correction? And we all decided that the only time you get DNA, that you can correct an error and identify an error is with intelligence? After seeing just that evidence alone, how can anyone not see that there's an intelligence behind what's inside of us?
Because their religion is more important than facts.
Thank you. That's it. That's it, right there. When you decide that only science that confirms what you believe is what you're going to accept, you've already stepped yourself out. It's hard to rationalize with those people. It's hard to have conversations with those people because they will deny the science that they don't want to accept and will accept only what they feel confirms what they already believe. This is a struggle I have on the internet. This is a struggle I have in conversations over coffee. It's a tough nut to crack. And it goes back to what we spoke about in one of our other talks. What these people have is not a head issue. It's a heart issue, right? I can give them all of this data and there will still be people that will walk away going, that's bunk. Junk science? Ah, that's junk science. You're just being crazy. We know, it is established that dinosaurs died out 65 million years ago. What do? There's no conversation to be had at that point because they're not open-minded, they're not free thinkers, and they're science deniers. I don't know where to go with that. At that point, I have to say, well, I would respectfully disagree, and then just move on. Maybe somewhere down the road they meet somebody else, or maybe I've left a rock in their shoe, and they think about it later and go, well, technically, I didn't want to admit it in front of him, but I don't know where that goes. I let the Holy Spirit do what he's supposed to do. As I said before, not my job. He walks away from me thinking that way. Okay. I don't know where to go with that. So you're right about that. And let's explore that since Gary brought it up. Let's talk about this just a little bit. So we've mentioned soft tissue. We've mentioned DNA, right? Now let's take it and bring it full circle. And let's really pile on the empirical evidence. Not historical stuff. Empirical evidence and see where it falls. So think about this. How many of you have or know about geothermal heating and cooling? Right? In this area, a lot of people know about that. What does that mean? It means you drill down into the ground and you drill over and you drill back up and you run these tubes and you blow air like this and it circulates through your house. What you're using is geothermal heating and cooling. Why do they do that? Because once you get about 6 feet underground, the Earth's temperature evens out, and it stays a pretty constant temperature all the time. So at about, and what, does anybody have a clue what that is? Like, it's about 55. 55 to 60, that's absolutely right. Okay? So about 55 to 60 degrees is what we get after about 6 feet underground. So, once again, out here on the surface, we'll get -10, we'll get 110 here in South Dakota, right? If we were underground, it would stay about 55 degrees-ish or so constantly, regardless of what the temperature up here was doing. Now think about what I told you about the bones themselves. The bones were buried how far underground? About 50 feet, right? So, and where were those bones? In rock? In what state? Montana. Does Montana get really cold in the winter? Does it get really hot in the summer? Yes, it does. It's our temperature, right? Keeping in mind that once we hit about 6 feet, 10 feet underground, it's all about the same. Now I want you to look at these rates based on what we just found out about geothermal. 55 degrees is approximately 15 degrees Celsius. 15 degrees Celsius on the chart tells us that the time in years that DNA will last is how much? 131,000 years done. At 131,000 years, you will no longer be able to detect DNA, according to a paper that was done on the decay rates of DNA and was the most advanced paper ever done on decay rates at the time. 131,000 years for DNA. Are we finding DNA in dinosaur bones? So they have to be, if we follow science and the truth, no matter where it leads, they have to be younger than 131,000 years, because at 131,000 you can't detect it anymore. So it has to be less than 131,000 years. Now tell me that doesn't blow a hole in their idea of 65 million years ago. Now if you're a young Earth creationist, you are loving this right now, because this would closer to confirm yours. But let me just remind you that even at 6 to 10,000 years, this is still 131,000, so we're not quite there yet. But it has to be less than 131,000. But I'll take 10,000 years compared to 65 million years. The difference is astronomical between the two. So we have soft tissue, we have DNA. We have DNA being found constantly over and over and over again. We have empirical studies on soft tissue. We have empirical studies on DNA. This is what it shows us. Those bones were found in Montana. They were found underground. They were found at approximately 55 degrees. I want to show you one more thing, and this is something that was brought up at the beginning of this paper that I did not get into. But the beginning of this paper even recognizes this. Let's see. I might not have it in here. This particular paper mentions, in the paper itself, it says that these decay rates are assuming, basically, that as soon as the organisms died, they were buried immediately. They even say we had to assume that these organisms were buried immediately at the time of their death, because we cannot account for how long they may have been above ground when they died. It's possible that they died above ground and were dead for an hour, a day, a few days, and they would have rotted a whole lot faster, right? Then they were buried? Well, you can't account for the decay rates, which would have been much faster prior to burial. And he says it in this paper. I'm just assuming that they were all buried immediately and there was no post-death decay at a faster rate than what I'm giving you here. So keep that in mind, right? Which just keeps adding to the problem here, which is what they found here, what we find in the dinosaurs could even be much, much, much younger than 100,000 years. We're finding DNA. Tony, you up there? Go back to where I was before. Take me to the slide right after that. If you would, please.
That one there, or keep going?
No, keep going. The one where I was, after the mosasaur, and it's here in town, and... Yeah, the next one. The next one. What's after that?
Going overseas?
Do that.
Got it.
Okay, so, here's something that... Yeah. All right, so, this is Thailand, this is Bangkok, Cambodia is what I want you to pay attention to, okay? In Cambodia, there is a building in Cambodia. Raise your hand if you've ever seen Tomb Raider. Lara Croft, Tomb Raider, if you guys want to admit to having seen it. I played the video game. I was wrapped up in the secular world for quite a while. I'll be the first to admit it. So I played a ton of Tomb Raider when I was growing up, except it was on like the computer and stuff, which you kids wouldn't know about because you have PlayStations and stuff. I don't get all the buttons. My brain doesn't work like that. I need the Atari, which was a joystick and a red button. And pound away at that thing. So this is one of the buildings down there, which is cool. Look at that giant. How cool, that tree that's growing out of the top of that thing. How cool is that, right? It kind of makes me want to go to Cambodia, but not really. Yeah, but not really. So this was a building that was used in Tomb Raider. They filmed down there at this place. And this is pretty cool because on the walls of this particular building, they've got pictures of stuff. This is in Cambodia. Can anybody identify what that thing is? It's not the best resolution, I apologize. Looks like a bird. Who said a bird? Someone said an ostrich. Would you think it might possibly be a flamingo? Can you see it? If we go back again here.
Kinda.
Can you go back one, Tony?
Got it.
There you go. Can you kind of see it now? Yeah, okay. So, anyway, on the wall of this thing, they have what is obviously a bird of some sort, maybe a flamingo or some kind of stork, maybe something. This is tougher. Can you guys figure out what this is? Looks like a bear. It kind of looks like a bear, right? But it's actually got something kind of coming out of its head, which makes a... Not an elephant, but you're getting warmer. Okay, so what if I showed you this? Like a water buffalo. It could be one of those other things too, but if we go back here, you can kind of see, now maybe it's kind of like a water buffalo, right? Kind of makes sense. This is on the same column, it's on the same wall, I think it's below the bird or whatever. Anyway, so it's a buffalo. What's that? Kind of looks like it, doesn't it? So it's a hippo, right? It's a rhinoceros with foliage behind it. No? Is that not convincing? Raise your hand if you think that's a stegosaurus. Okay, raise your hand if you think that's a rhinoceros, or like a rhinoceros with just trees behind it, like foliage or plants behind it. Maybe? Do you see it? You guys kind of see it? So there are a couple that do see that. I don't know. I think it is what it is, right? I mean, it certainly looks like a stegosaurus, does it not? It looks like a dinosaur. This is on the same exact column as the bird and the water buffalo, and then right below it is this thing. How would they have known what it was if they hadn't seen it? How would they possibly have known had they not seen this thing? YouTube, right? 5,000-year-old YouTube. You betcha. It is, that temple, I believe, was 3,500 years old, 4,000 years old, something like that. Very old. Very old. So, yeah. So this is what's on the exact same. Now, what have they done with this? They tested it. Like, in between the cracks, you see all the weathering and stuff. That's called patina. They take that out, and they can actually test that from a molecular perspective and test it against the rest of the other animals that were pictured on the wall. and they all match, and they all come back at about the same date range. It's also at the same level. So if you look at the wall, it's all flush with the different birds, which means something here wasn't scratched out, and then they carved a different animal in instead, just to kind of make it look like there's a dinosaur there. No, it's all flush. So they know that hasn't been done. So this is legit. Like the people that made the bird and the water buffalo, they drew this at the exact same time as they drew the other stuff. This is in Cambodia. Now let's go up to Kachina Bridge. This is in, you see Colorado, this is in Utah. And up on the side of this cliff, it's kind of a mountain, it's like a little rock wall there. You see this. Can you see this? It looks like the Sinclair Station dinosaur, doesn't it? That's called a sauropod dinosaur. That's your Brachiosaurus, Brontosaurus, and those types of dinosaurs. This is on the wall. There's also what appears to be kind of like a man there. This is all this little rough stuff, this colored stuff is patina. They've tested it, and it all matches around here and around here, and it all comes back at about 800, I think, AD. And that's about the time that they had natives living in this particular area. They lived in the rock wall and stuff. They lived in caves in the rock walls. And these were part of the pictographs that they found on the walls themselves down in Utah. You have to take a ladder with you to get to it. You put this big long ladder, and then you got to crawl up the side. And when you do, this is what it looks like originally. So that was the enhanced version I gave you before. But you can see what it looks like now. And they've tried to take this dinosaur and pull them apart and say it's actually 3 different pictures and whatever else. But I'm just asking you to make up your own mind. I think it's pretty simple to see what it is. It's pretty clear. So we have Cambodia, we have Utah. Now let's go to Carlisle Cathedral. This is over in Ireland. So in Ireland, they have a place called Carlisle Cathedral, and if you walk in, there's a rug on the floor. And if you move that rug aside, you'll find some brass pictures, and this is there. And I'll give you the enhanced version next. You can see down below the enhancement. It's 2 dinosaurs. Now, it's very interesting. Someone said dinosaurs fighting, right? Okay.
I don't think that's funny.
Okay, this is funny because you could be right. It's kind of one and the same. It's actually what they believe to be a mating ritual, which is common. Sometimes you fight and it all works out in the end. But this is what they see out of giraffes nowadays. This is how giraffes, they flirt with one another. They wrap their necks around each other, right? And so this is drawn on the floor in brass of Carlisle Cathedral in Ireland. So this is a very recent picture that was drawn of what certainly appears to be sauropod dinosaurs. Here's an interesting note. Take a note of this tail here and this tail here, right? These are very different, correct? Now, so when non-theists looked at this, they would claim that they're clearly not dinosaurs. They're probably small lizards of some sort. Because dinosaurs did not, especially sauropod dinosaurs, did not have these club tails. They had these kinds of tails, but not the club tails. So that was word to them that it was some sort of other reptile or something like that, and that's what the picture of it is. However, this is great. Just a few years ago, they dug up a sauropod dinosaur, and guess what its tail looked like? It had a club on the end. So they just recently dug up a sauropod dinosaur, and it had a club on the end of its tail, which they had never found before. So, pretty funny. So yeah, so now we're Cambodia, we're Utah, we're Ireland, right? Well, this one can't be 1600s, 1500s. So we're getting closer to modern day, right? Pretty crazy. So this wasn't too long ago. On a side note, this is for the kids. Dinosaur, the word dinosaur is actually a newer word. Dinosaur wasn't always dinosaur. It came about in early 1800s or 1700s. Do you know, Mike, what that date was by any chance? You're pretty smart. It's like early 1800s or 1700s is when the guy actually coined the term terrible lizard, which was dinosaur. Okay? Before they were called dinosaurs, people knew about them, but what did they call them? Do you guys know? Dragons. They called them dragons. So you can go back in Old English literature and it talks about her, so Sir Edward or whatever slayed the dragon, and everybody thinks, oh yeah, haha, it's fanciful, mythological, fire-breathing dragon type thing. No, maybe not. It's possible, because that was the word for a dinosaur back then. It's possible that what he slayed was a dinosaur. I'm stretching it a little bit, but take it for what it's worth. Just know that dinosaur is a newer word, 200 years old or so. And then, this is what it would have looked like. Yeah. And then, if we go down to the Amazon, this was a recent find. This was just found in the last 5-10 years or so. Another pictograph on the side of a wall, and what is that? It looks like a sauropod, right? It looks like another sauropod dinosaur. And look, and there's like 9 hunters around it getting ready to kill it. Must have been pretty big to have 9 hunters after it, but yeah. So once again, Vance Nelson is the guy that brought this to light. Untold Secrets is his website and his book. He's got a book out where I pulled a lot of that information from. I had heard it, but he put it all in one spot that was easy to get to. And he went down there and looked at it, and then prior to releasing any of this, he spent like 3 to 5 years studying it. And patina, and they tested the wall samples and the whole bit, because here's the deal. You come out with something like this as a Christian or a creationist and you get it wrong, forget it. Forget it. You'll never talk anywhere again, ever. In the secular world, they'll just, you're done. Remember that whole thing with the Amazon dinosaur, blah? Yeah, you're discredited. So we have to be very careful with any kind of science that we do that's built around this kind of stuff, because if we get it wrong, we're going to get hung. We're going to get hung for it. So with that brings us actually to the end of the presentation itself. That was a little... So the evidence is out there, right? Like, it's what I showed you tonight. I want you guys to keep this in mind, okay? What I gave you tonight was secular, peer-reviewed research. I didn't send you to go to this weird Christian website. And that's not to say that Christian PhDs don't do good science, because they do amazing science. And I love what they do because they come at it from a different angle than the secular scientists do. They come at it from a, we are created, let's try to examine the body as if we were made by a creator. Whereas the non-secular scientists, or the secular scientists, come at it from the view of, we were not created, we evolved over millions and millions and millions of years. And so their whole, how they approach science is completely different than how a Christian scientist approaches science. And because they do what they do, they get dogmatic in their thinking, and they miss stuff that would be very, very interesting, I think, to the rest of the world, that they would just bring it to light. So what I showed you tonight was secular peer-reviewed research. It's all out there. This isn't something that was created and put out, you know, blah, blah, blah. This is in the secular review. They just read it differently. And you saw Mary Schweitzer, like when she talked, she's like, man, this bone's 68 million years. We don't get it. We just don't get it. So they're very dogmatic in how they think. But I wanted to bring this to your attention. I wanted you guys to see this because it really actually helped me in my journey. It was just really interesting to see this empirical evidence that's out there for dinosaurs. And what you do with it is what you do with it. But I think it points to the idea that these things maybe were around even a few thousand years ago. As crazy as that sounds. And I'll be honest, when I'm talking to people, I don't open with that. Hey, did you know dinosaurs were around 2,000 years ago? I don't open with that because people are like, yeah, never mind. But the science is there. So when you're having discussions with your friends and your family and your kids and whatnot, let them know that there's lots of really strong science, and it's all built around this soft tissue. It's all built around what we saw tonight. We have the actual decay rates of soft tissue and DNA, and we're finding soft tissue and DNA in dinosaur bones over and over and over and over again. And they keep pounding the drum. 68, 110, 80 million, 150 million years old. They keep pounding that drum, but I want you guys to be well educated. I want you to know that that is not what the science shows. The science shows that they cannot be millions of years old. So with that, do you guys have any questions? Anything else you want to bring up?
How old is the earth?
Well, that's a very good question. I would say I would lean towards 6 to, I'd maybe give it 12. I'd say about 6,000 years old. That's where I would put it. 6 to 12 maybe. That's where I lean. And we've talked about this before. I think there's a lot of good evidence for an old Earth and an old universe. But once again, I think you guys would be amazed at the evidence there is for a young Earth and a young universe. It's crazy when you see it. Once again, it's all out there in the peer-reviewed literature, yes.
So Hanna-Barbera had it right with Flintstone, yes?
Well, yeah, kind of. I don't know. I don't know if they had little pedal cars back then or not, but yeah, maybe it's possible. I will say this about, I'll make this really quick note. Speaking of the cavemen and stuff, cavemen are typically supposed to be really, really stupid. Club the women over the head, drag them back to the cave. You guys have kind of heard that. Maybe you've seen that. We know that is absolutely not true nowadays. The fact that they lived in caves doesn't make them any less or any less stupid than anybody else. Living in caves is actually really smart. Why? Because the temperature is constant. There was one way in and one way out, which was perfect for predators and enemies. They could only get in one way and they could only get out one way, so it was easy to defend, it was easy to protect. You let a fire in there and you could warm the whole place. And they've now found these places where Neanderthals lived, and they did different things in different places within the cave. In this room they made food, in this room they made jewelry, in this room they washed clothes, in this room they cleaned the food and got ready for that night's thing. They had different rooms that did different things. It wasn't any different than what we do today. They didn't do it in a house. They did it in a cave. And they also made glues, really strong glues that we can't in today's world duplicate. They buried their dead. Like I said, they made jewelry. So these Neanderthals weren't stupid. They were really not much different than you and I. And they have found Neanderthals buried with Homo sapiens, for those of you that want to take that next step, because they thought that, remember we talked about what a species is and what a different species is, right? They thought that Neanderthals and Homo sapiens were different species, and now they know they weren't different species. They were mating and having children, and we are products of that because we all have DNA now, Neanderthal DNA within our genomes, and this is what they're finding when they sequence modern day humans. They're finding Neanderthal DNA, and that we find Neanderthal bones and human bones all buried together, so we know that they coexisted with one another. And the more we learn about science, it just starts all kind of coming together, right? It's really interesting how it all works. Any other questions?
Are you going to start apologetics meetings on Saturday?
Oh, South Dakota Apologetics. Yes, I'm part of South Dakota Apologetics. We will be starting up those Saturday morning meetings again, probably, where are we at, August, September time frame. We'll probably kick those off again. If you guys like this kind of stuff, this is kind of what we talk about. It's not always science. As a matter of fact, it's rarely science. We talk about other stuff like this type of evidence for the Bible, this type of evidence for the existence of God, like really deep, really intellectual type stuff, but nothing that you can't follow. You guys would all understand it. It's just a little deeper step than what most people talk about in a typical church. So yes, probably in September. We meet at Starbucks downtown in the Alex Johnson at 9 a.m. on Saturdays, for those of you that want to pop in every now and then and maybe get your fill of something. We just finished up a study on the historicity of the Gospels, actually, which was a very interesting study. Anything else? I want to thank everybody for showing up these past few weeks. Once again, it's been a huge blessing for me to be a part of this. And you guys I figured if we get 10 people to show up, that would be cool, right? And we got all these people to show up, and not only that, but you kept coming back. Like, it wasn't just 1 week or 2 weeks and you're like, never mind. Like, you kept coming back. So I hope you learned something from all of this. I hope you were all blessed by all of this. I have packets that I made up for you guys, if you're interested, on tonight's talk. And this is the peer-reviewed research we went through, and I give kind of an outline of what to look at in these particular sections. So I think you guys, if you like what you heard tonight and just want to have something to hold on to, this is it, right? This will walk you through kind of the entire talk. The copies aren't necessarily the best, but if you need something, you can see the title and you can find those things online and look them up for yourself. I always tell everybody this, don't take my word for it. Prove me wrong. Yes, yeah, prove me wrong. Check me out, okay? Go look this stuff up online and see if I misrepresented anything I gave you here tonight. Look it up and see if I'm trying to angle this from a particular view because I'm a Christian and I want you to believe a particular way, so I'm angling the evidence from a particular way. Check me. Look that all up. If I'm misrepresenting anything, you come back and you tell me. I want to know because I want to fix it. I want to tell you guys the truth because I know where the truth leads. I know where it goes. Jesus Christ came to earth. He lived the perfect life, the only person ever to do it. And when he lived that perfect life, he chose to die on that cross. For you, for me. The punishment that I deserved, the punishment that should have been rained down on me, was put down on him. And he did that for me so that one day I can be with him in heaven. That's the good news of Jesus Christ. The bad news is I'm a sinner and I'm not perfect and I never will be. And I will sin tomorrow and I'll sin the next day and I'll sin the next day. But when you recognize you're a sinner, when you ask for forgiveness for those sins, when you tell Jesus, I am sorry for all that I've done, he takes every one of your sins, the pages and pages and pages of it, and he goes, okay, let's start again. This is what Jesus did for all of you here. All you have to do is put your faith in him. Recognize he walked this earth. Recognize he died on the cross. And most important, recognize he rose again 3 days later to prove to everyone around him that he was who he said he was. I am that I am. I am God. And he proved it to everybody. He stayed on earth another 40 days, and he talked to thousands of people after that to let them know, look, I'm back. And not in spirit form, in human form. Go ahead, touch my hands. See where they drove the nails. He did this for you. If by chance you've never ever given your life over to Christ, I want you to think about it. This life is not our home. Rapid City's our temporary place of existence. What we want, what we're looking forward to, is the next place. When you die, your body, it will decay. The DNA will decay. The soft tissue will decay. But your soul will not. Your soul is immaterial, and it will live on forever. And while you're here on earth, you choose where you go. God doesn't send you to hell. You choose to go to hell when you don't choose him while you're here on earth. You either choose God or you choose not God. And if you choose not God, when you die, because God loves you, against his better wishes, he gives you what you asked him for. And that is not God. It is a place of eternal separation from God that we Christians call hell. It's everything heaven isn't. If heaven is all fellowship, there is no fellowship in hell. If heaven is no pain, it is all pain in hell. If there is no suffering in heaven, it is all suffering in hell. And that's where you reside, not for a year, not for 2 years, not for 80 years, but for eternity. That's the deal. I don't make the rules, I just live by them. So if you have not given your life to Christ, I want to challenge you tonight to take the evidence that I've given here tonight, the evidence I've given you over the last 5 weeks, and I want you to think about where it points. I want you to be open-minded. I want you to open up your hearts. I want you to open up your minds. It's all I've ever asked of you since we started this whole thing. Follow the truth where it leads, because I promise you, after decades and decades of studying the evidence, it only leads one in one place only. It always falls heavily on the side of A, theism, and B, Christianity. Period. Now, how you do that is up to you. I'm not into the sinner's prayer. I'm not into say this prayer and voila, you're saved. I don't do that. That's for you to decide. You go home tonight, do it tomorrow, however you want to do it, in your way, in your words. And just tell Jesus, I recognize I'm a sinner. And I need a Savior, because I cannot get into heaven on my own. I need your help. Forgive me for the sins that I've committed against you. And if you just do that, Christianity is the only religion in the world that is not works-based. You don't have to do any works to get into heaven. God says, here's a free gift. Just take it. Just reach out and take it. I don't want anything in return. Just take this free gift I'm giving you. And if you'll do that, you will beat death. It is the cure to cancer. It's the cure to pain and suffering and agony. A free gift. He asks nothing in return. Just put your faith in me, because I will be here when you're gone. And you will be with me forever. I want to see you all there. I don't care how you got to get there. I don't care what you have to do. But you get there. It's the words I say to a lot of the people I meet and all the people I love. I don't care how you get there, but you get there. I want to see you there someday. We're going to have more conversations. There's a lot more to have, because this isn't it. Let's pray. Heavenly Father, we just want to thank you for your mercy. We want to thank you for your greatness. Before I came here tonight, there was a song that was playing, and it said, I praise you for your mercy that leads me through valleys of sorrow to rivers of joy. From valleys of sorrow to rivers of joy. That's what you get. God makes everything good. If you've got something in your life that's bothering you, if you've got something that's hurting you right now, God wants that. You need to give that to him. If there are struggles in your life right now or something you don't want to give control of, God won't force you to give it to him, but he's going to tap at you and tap at you and tap at you, and the Holy Spirit will wear you down if you don't just turn it over to him. He wants your burdens. He wants your problems. If you just have faith in him, just give it to him, he will take care of it for you. Valleys of sorrow, rivers of joy. That's what my Savior gives us. That's what our Savior gives all of us. I pray minds were open. I pray hearts were open. I hope the information that was given was to your glory, not to my own glory. I want people to better understand you, Lord, to know you're real, to know it's true. We can look at it from every single angle, and you are there everywhere. And we thank you for that, and we praise you for that. And I thank you for the blessing these past 6 weeks have been for me. I ask this all in Jesus' name, amen. Thank you. I have packets.